Hello, everyone, and welcome to this podcast. And in this podcast, I'm going to take a few moments to go over some of the activities that we did in class the other day. I made an error that I mentioned in class, and then I corrected it, but I wanted to make sure that I went over that activity again the correct way so that you had it correct in your notes. So this was the activity that we drew a segment of DNA with two genes on it going in different directions. So let's begin by drawing the DNA here. 5' to 3' on the DNA, and then 3' to the 5' on the DNA down here. And the instructions for the activity says that gene A should be read on one of the strands, or on the top strand. So I'll put gene A up here, and gene B will put on this strand, or reading from this strand. Let's start with gene B. Remember, RNA polymerase is going to read 3' to 5', and so it's going to read in this direction. Gene B will occupy this area, we'll say. So we'll put a B here. And this area here we'll call the ORF, the open reading frame. It's called the ORF, or open reading frame, because this is the sequence that will ultimately make the protein. How does the RNA polymerase know to start here to make the RNA? It knows that because the promoter is just upstream of the open reading frame. And it knows to stop making messenger RNA because there's a terminator sequence here. When we draw gene A, we'll also have a promoter and terminator. Each gene has to have its own promoter and its own terminator. Now, as the RNA polymerase, and I'm just going to show it here in a dashed line so it doesn't take away from what we've already drawn. And remember, it's going, in this case, from left to right, 3' in to the 5' in. And this is where I made a small mistake yesterday in class that led to some confusion. So I want to describe it in a slightly different way. Remember, the RNA has to be anti-parallel with this strand. So in the end, it's going to look something like this, with a 3' end here and a 5' end here. And so the RNA polymerase is chugging along this way, 3' to 5'. First nucleotide that comes off is going to be 5'. And so if we do a little time-lapse photography here, this will be 5', and this will be 3'. So as the RNA polymerase moves along here, the new part here is forming here, so it'll be 3', and this old part down here is the original 5' end that was originally sequenced with the first nucleotide. If it helps, you can think of it as like the RNA polymerase is moving along here, and this string of RNAs extending from it almost looks like a cape, if you use your imagination, right? Then, when it's all done, it's all the way to the terminator sequence, then this will be released, as we had drawn here originally. And now, in bacteria, this can go straight on to make proteins. We'll learn in a little bit that if it's eukaryotic, some additional things have to happen to this RNA. Now, let's move to gene A, and let's make gene A here. And remember, it's read this way. So the promoter will be on this side of gene A. And so it knows when to terminate transcription, it has a terminator. Our RNA polymerase will recognize the promoter and begin making RNA. So as it's moving along here, the first one is going to lay down right like so. The end here, because it's going this way, this end's going to be the 5' end, and this end will be the 3' end. As it moves along, it's going to release the RNA originally made, so a little time lapse here. This 5' end here, which is anti-parallel with this one here, this end of the DNA, extends out. The 3' end right here is where the new DNA is being added. Again, it extends out, kind of like the cape for the RNA polymerase. When it gets all the way to the end, just like we saw on the one below, it will release the whole RNA, 3' to 5', anti-parallel with this DNA strand. Now the last thing we need to show on here is the template and non-template strands. The template strand is the strand that's red, so for gene B, this is the template. For gene A, this is the template. The template will not be the same for every gene, and the non-template will not be the same for every gene. So the non-template is this strand, and sometimes we call that the coding strand. The next questions on this ask what would happen if the promoter is mutated, the open reading frame is mutated, or if the terminator is mutated. Remember, if the promoter is mutated, the RNA polymerase cannot bind. If the RNA polymerase cannot bind, it will not be able to make messenger RNA. So no messenger RNA and no proteins would be made. If the open reading frame is mutated, the promoter is still there, so the RNA polymerase will still bind. It's still going to make RNA from what's left of the open reading frame. It'll still terminate, so you'll still make an RNA molecule, but it'll be smaller, and ultimately it will make the wrong protein. If the terminator is mutated, you'll still make RNA because the RNA polymerase is still binding to the promoter. Open reading frame is fine, so you're still making that. However, the RNA is going to be much, much larger. because the terminator's not there. Again, you still have the messenger RNA, and it still has the potential to make the protein. But depending on how long the RNA gets, you could imagine that that might affect how the protein will be made or whether or not this RNA will survive. All right, this is all I have for this activity that I made an error in during class, so I want to make sure you have this. I also rushed through the second activity because we were running a little bit behind. And so, I'm sorry, the third activity. And so I want to go over that again, just so you have the correct answer. Now, this activity asks you to think about termination of transcription. So here, let's draw our gene here, or our DNA here, with our 5 prime, 3 prime, 3 prime, 5 prime, ends as so. And in this case, we're just going to talk about one gene. So let's make our open reading frame for gene A right here, and then the terminator here. And now we're specifically talking about bacteria. And we're going to have it reading this bottom strand, so it's going to go this direction. And so we have our promoter, and so we have the RNA polymerase here, moving along here. And if this was row dependent, as it gets here towards the terminator, and we'll go ahead and draw our RNA coming off here. In row dependent, remember we have this row factor binding here, and remember row binds to a sequence here, initially at least, that we call the rut site. And its job is to terminate transcription, but it can't, because it can never catch up with the RNA polymerase. And I guess to be accurate, we should put the RNA polymerase here because it's been moving along. But what happens is we have part of the RNA here near the terminator that forms this stem loop. This stem loop, and we're not really sure how this happens, but it somehow or another causes this RNA polymerase to pause. And that gives it enough time to move along here with its helicase function and buzz saw through these hydrogen bonds here to release the RNA. Now the question asks, what happens if we change A's and T's that might have been along here in the terminator to G's and T's? So A's and T's in the DNA sequence to G's and C's. If these are A's and T's, they're weaker bonds, and A would pair with a T and T with A's, of course. With only 2 hydrogen bonds between each of those, it's easier for it to go through. However, if it changes to G and C, it's more difficult, but a row is still able to handle that. So if you make this mutation in a row-dependent termination strategy here, should see no effect, so no problems. But now let's look at row-independent. Everything else is pretty much the same with a row-independent, so let's write row-independent. So row's not going to be involved. So I said most things are pretty much the same, with the exception that there is no row. In row-independent, remember, you still form this stem loop, and this stem loop somehow or another, again, not completely sure how, but it somehow or another interferes with RNA polymerase's ability to move forward, at least at a decent speed. And the way that the RNA molecule here is going to fall off is because there are very weak interactions here at the terminator region. You have a series of A's and T's. You have a series of A's and T's here on the DNA that are going to make corresponding U's and A's on the RNA. These weak interactions aren't strong enough to remain bound as this RNA polymerase stalls, and so it will fall off. How exactly it falls off, we're not sure, but it does. And we know if we change these A's and T's to G's and C's, that interaction is too strong. Too many hydrogen bonds. And so even if the RNA polymerase pauses, it's not enough to allow this to fall off. Again, we're not sure how it falls off under the normal circumstances, but we know if we increase the number of hydrogen bonds here, it can't fall off. So for row independent, if you have mutations that convert A's and T's to G's and C's, you will prevent termination and it will just stop here. All right, so I'm sorry I rushed through that in class, but I did want to make sure you had the correct answer and explained well enough so that you can prepare for the exam. If you have any questions about these 2 activities or any activities that we've talked about, please make sure you stop by. If not, I'll see you in class. Bye.